So hello everyone, welcome back to my channel. So today in this video we talk about the topic that is lab Operon. So it's actually a very interesting topic. So let's start from the beginning. Okay, so first of all I write here the lab Operon. Okay, lab Operon. First we have to understand what is the meaning of this word, right? So lab word comes from the word that is lactose. All we know the lactose. Lactose is one type of sugar which is also present in milk, right? Now the question answer, what is Operon, sir? If I say simply in a definition that it is a cluster of genes which have common promoter and operator. So what do you say? I just will not understand here. Okay. Actually, we all know that in our whole body made up of cells, right? And in every cell all have the genes. But which gene functions where? Or you like to say which gene express in which region of our body, all controlled by a switch, and that switch is called Operon. Okay, Operon is like a switch. Suppose in your classroom there is a switchboard, right? And this switchboard, if you on the switch, the fan is on. That means how the fan control? The fan controlled by a switch. If you on the switch, fan is on. And there is also a regulator, right? If you regulate the switch, you can control the speed of the fan, right? In the same way, if we regulate the gene, all the gene regulated by a switch, and that switch is called Operon. Operon is like a switch which controls the different type of genes. Okay? How it functions? There are actually same type of gene. Same type of gene also present in our eye, same type of gene also present in our hand, same type of genes are present in our chest. But which gene, same gene, in which area, how it's work, how it's controlled, when it expressed, all controlled by Operon. Okay? Now the question arises, how this concept comes? Actually this concept comes given by two scientists in 1961, that is Jacob and Monon. There are two scientists, they also given this concept. Actually they have done some experiment. Okay, they are taken two medium. One medium, suppose this is A, this is B. Okay, suppose in the medium A, they also given the potato. Okay, all we know the potato. Potato is a high rich of carbohydrate. Okay, and in the medium B, they have also given milk. Okay, so in the first medium A, they have also given bacteria. Suppose E. coli bacteria, they are given. They observe there the E. coli bacteria here grow rapidly. So how they grow? So we know in the potato we also have glucose, right? It also have glucose because this medium is glucose rich, right? And from this glucose the bacteria get their energy. So that's why here they grow rapidly. But in the same type of medium B, they have also given the milk. We all know the milk also have present lactose. There are no present any glucose. It's also present have lactose. So if same bacteria also given in this medium also, okay, then observe shortly that here bacteria also will grow rapidly. Now the question arises, how the bacteria get energy from the medium B, because there is no glucose, there is lactose present. Then they observe that the lactose, somehow they break down the lactose to form glucose and galactose, and from this glucose they can get energy. So that's why here bacteria grow, but slightly slower than the previous medium. Okay, so from this experiment they all concluded that the bacteria have some gene also present within their genome, which are responsible from the breakdown of lactose. From this lactose they can get glucose. If there is a situation where the glucose is not present, in that time the operon is on, in that time they break down the lactose, then they can get glucose. Okay, they all proved from this experiment. So how this bacteria break down the lactose, how the lac operon works, we can go through from the next video. Okay, so I think all of you understand all this topic. If you understand this topic, please like my video and please subscribe my channel and please stay with us. Okay.